So now we're going to get into the instruments of microscopy. So we're going to talk about a lot of the different types of microscopes here. The early pioneers of microscopy opened a window into the invisible world of microorganisms. We've talked about that. But it continued to advance in the centuries that followed. So 1830, Joseph Jackson Lister created an essentially modern light microscope. So basically what it is that we know as a light microscope today. The 20th century saw the development of microscopes that leverage non-visible light, so using some of those other EMR, and we'll see those in a moment, such as fluorescence microscopy, which uses ultraviolet light source, and electron microscopy, which is the short wavelength electron beams. So actually taking electron beams and hitting an object, and then that beam bounces back and we can see things in much, much more detail. These advances led to major improvements in magnification, resolution, and contrast. Remember, those things are things we need to manipulate in order to see an object very clearly. By comparison, the relatively rudimentary microscopes of Van Leeuwenhoek and his contemporaries were far less powerful than even the most basic microscopes we use today. So here we are, we're going to take a look at these different types of microscopes. We'll start here with light microscopy. So many types of microscopes fall under the category of light microscopes, those which use light to visualize images. So when we talk about light microscope, very clearly it's just using light in order to see this image. Examples of light microscopes include Brightfield microscopes, dark field microscopes, phase contrast microscopes, differential interference contrast microscopes, fluorescence microscopes, confocal scanning laser microscopes, and 2-photon microscopes. You need to know those things. Okay, we're going to talk about each one of those, and it's important that you know that these are the different light microscopes. And we're going to talk, and as we talk about each one of these types of microscopes, it's important that you understand the concepts behind each of them, why they're different or how they're different, and why they're used for what they are used for. So first we'll start with our Brightfield microscopes. So the Brightfield microscope, which is perhaps the most commonly used type of microscope, is a compound microscope. Remember, a compound microscope is one that has more than one lens. So we have the simple microscope, which is a one lens microscope, one of the first microscopes. And then we have compound microscopes, which has two or more lenses. And they produce a dark image on a bright background. So bright field, the field of the area is bright, and then the image on it is darker. Some bright field microscopes are monocular, which means that they have a single eyepiece. You can kind of see the image down here. This is the eyepiece. The ones in the SC4 classrooms at the moment, most of them, are monocular, but most newer bright field microscopes are binocular, having two eyepieces, as in this image here. In either case, the eyepiece contains a lens called an ocular lens. So you can see in here, there's, well, it's not on that side actually, but there's a lens in here that's called the ocular lens. The ocular lens typically magnifies images times 10, and that is how it is in the classrooms at SC4 as well as other campuses. Most times the bright field microscopes that are used in a laboratory oftentimes are 10x in the ocular. At the other end of the body tube are the set of objective lenses. So we're going to scroll down a little bit so we can talk about these things and see the image here. So up here, objective lenses on a rotating nose piece. So you can see objective lenses. These are objective lenses where you switch between generally the 4x, the 10x, the 40x, and then the 100x. And it says that right here. The magnification of these objective lenses typically ranges from 4x to 100x. And it's indicated on the lens itself. You can see the writing here. And then this right here, you can see it's labeled, is that rotating nose piece. So you can kind of click those lenses into place. The ocular and objective lenses work together to create the magnified image. So this is where the compound microscope part comes in. We have a lens in here, and then we have the lens up here. So these two lenses working together give us our total magnification. The total magnification is the product. If you remember some math classes, product is multiplication. The product of the ocular magnification times the objective magnification. So ocular magnification, oftentimes 10x. The objective lenses, remember, are anywhere from 4x to 100x. So depending on the objective lens that is being used, you take that objective lens number and multiply it by the ocular lens number to get your total magnification. Here you can see the example. If you're using the 40x objective lens, and then you have your ocular lens of 10x, you have the 40 times the 10, giving you 400x. That's the total magnification that you're using at that moment. Down here, the item being viewed is called a specimen. The specimen is placed on a glass slide, which is then clipped into place on the stage. And so you can see the stage is labeled here, number 6. This whole thing right here is 6. The stage, and you can see actually right in here is where the clip is. So the slide will sit right under here and be clipped in. Once the slide is secured, the specimen on the slide is positioned over the light using the XY mechanical stage knobs. So these right here, XY mechanical stage knobs, you can kind of see them right here. And that's going to move the slide on the stage. So this way, and then kind of this way on the stage though. So these knobs move the slide on the surface of the stage, but do not raise or lower the stage. Once the specimen is centered over the light, the stage position can be raised or lowered to focus it. The coarse focusing knob, or the coarse adjustment knob, you can see right here, is the larger knob. You can see as number 4, the outside knob, this big one here, is our coarse adjustment knob, or coarse focusing knob, used for large scale movements. So that's only with the 4x and 10x objective lenses. And that's very important that that's only ever used with those two types of lenses, because if you use it with the 40x or 100x, you can run the chance of smashing the objective lenses into the glass slide and break either of them. Most often it's the glass slide that's broken, but it can also damage the lens. And then that's a really expensive fix and can mess up a lot of viewing after that. The fine focusing knob, which you can see labeled here, number 5, which is kind of within the large focusing knob or the coarse focusing knob, is used for small scale movements, especially with the 40x or 100x objective lenses. So this is when you especially want to use the small one. And that is when you use only the small one, is when you're at 40x and 100x. If you use the bigger one, again, you can smash the slide, but then also it actually makes it really difficult to find the object. It moves so fast, it can get out of the field of view right away. When images are magnified, they become dimmer because there's less light per unit area of image. So even when you have a really bright light down here, if you are at 4x and 10x, you may think it's really, really bright. It's hard to see because it's just so bright when you're looking at it with an eye, or with your eye. And then when you get down to the 40x or the 100x, you might be thinking, gosh, this is really not bright and something happened to my light. But it's just because you're focusing in smaller and smaller and smaller area, and so there's less light per unit of that area. Highly magnified images produced by microscopes therefore require very intense lighting. So things that can focus in even more need a really intense light in order to get in and make sure that there's enough light in there. In Brightfield microscope, this light is provided by an illuminator. So down here we have labeled number 7, where our light is coming from, is the illuminator, which is typically a high intensity and light bulb. Light from the illuminator passes up through the condenser lens, which is located below the stage. So here is the condenser lens. You can see it's mentioned as the condenser. And that focuses all of the light rays on the specimen. So it's taking the light that's coming up from here, and it is grabbing it and kind of pulling it into middle area here, and it maximizes illumination. So it's trying to pull in as much light as possible to get it through the specimen. The position of the condenser can be optimized using the attached condenser focus knob. So there is a focus knob up here that can allow you to move the condenser to try to adjust that to make sure you have optimal light. Once that optimal distance is established, the condenser should not be moved to adjust the brightness. So even though you may be moving up to a higher magnification, then at that point you don't want to change that. It's just going to, again, it's going to be a huge switch when you're looking at something at those higher magnifications. Doing even something tiny with some of the parts of the microscope really makes a huge difference because you're so far zoomed in. All right, so if higher levels are needed, the amount of light striking the specimen can be easily adjusted by opening or closing a diaphragm, which is between the condenser and the specimen. So we can scroll back up here so we can see where the diaphragm is. Again, right under the stage, so right here, actually up here is the diaphragm, and that also can open and slide open, slide close to then adjust the light that's coming in. And in some cases, the brightness can also be adjusted using the rheostat, which is a dimmer switch that controls the intensity of the illuminator. So it is making the light bulb on the bottom brighter. Here's the rheostat in this one right here. Can make the light down here, remember the illuminator, brighter or not so bright, a little bit more dim or a little bit brighter. A Brightfield microscope creates an image by directing light from the illuminator, so the light bulb at the bottom, at the specimen. This light is differentially transmitted, absorbed, reflected, or refracted by different structures. So based on the specimen itself, it's either one of these things is going to occur. Different colors can behave differently as they interact with the chromophores. So chromophores are pigments that absorb and reflect particular wavelengths of light. So again, when white light is coming in, white light, for example, coming from the light bulb, comes in and it hits something. So when it hits a prism, for example, it'll scatter that light into the different wavelengths, or because of the different wavelengths, and we see that as different colors. When we are talking about chromophores, they're pigments that are in a specimen, in different parts of the specimen, and they either absorb or reflect different wavelengths of light. So remember, if it absorbs that wavelength, that's the color you do not see. And if it reflects a particular wavelength, that's the color that you do see. Often, chromophores are artificially added to the specimen using stains. And again, remember, those are going to increase the contrast and the resolution. So we stain different specimens to make sure that we can see them compared to other parts of the specimen, but then also because it gives it different colors depending on what you're staining. In general, structures in the specimen will appear darker to various extents than the bright background. So again, it's a dark specimen on a bright background, creating maximally sharp images at magnifications up to about 1000x. And remember, that is what most of the microscopes go up to, is 4x to 100x, but then you have to take into consideration the ocular magnification, which was the 10. Further magnification would create a larger image, but without that increased resolution. So you're not going to get that resolution with the Brightfield microscopes. But this allows us to see objects as small as bacteria, which are visible at about 400x, or that 40x ocular or objective lens, but not smaller objects like viruses. At very high magnifications, resolution may be compromised when light passes through the small amount of air between the specimen and the lens. So when we're still talking about Brightfield microscopes that go up to the 100x, but when you're at high magnification, so when we're getting up to the 40x, but also the 100x mostly, the resolution may be compromised. So we may not get as clear, right, as high of a resolution when light passes through the tiny amount of air, the small amount of air that's between the specimen, between the slide, and the lens. So there's a little tiny amount of air between the two. Let me scroll down, you can see this here. So we're talking about this tiny space in here between the slide, which is here, and then the lens, which is right up here. And this is due to the large difference between the refractive indices, remember the refractive index of air and glass. Remember the light waves are going to move different between the two. And since there is that difference, remember it's going to refract. The air scatters the light rays before they can be focused by the lens. And they're showing that here in B. So they're showing on this side without immersion oil, which we're going to talk about in a moment. So we have our light source here, and we have our microscope slide with our specimen here. And when we get up to the 100x, the lens is so close to that specimen that when the light is going through the slide, through the specimen, then it hits the air, it's going to bend it so much, refract it so much, that you really can't get that resolution. To solve this problem then, a drop of oil can be used to fill that space between the specimen and an oil immersion lens, which is the 100x. So with the immersion oil, we can see here, we have the light coming up this way. We have our microscope slide here with the specimen. We still have our lens, but now we've got that drop of oil in here. And what that does is the oil, actually it should say it up here, yep, here, since the oil has a refractive index that's very similar to that of glass, it increases the maximum angle at which light leaving the specimen can strike the lens. So rather than the light coming through the glass and then having such a huge difference when it gets into air for the refractive index that it's just going to start going all the way out here and we can't really see it, instead, if we have this oil, it creates this barrier. So there isn't any air for the light to go through before it hits the lens, and then it continues on a very similar path as the one it was going through when it was going through the glass slide. This increases the light collected and thus the resolution of the image. So a variety of oils can be used. All right, so here, this is very important for those working in a laboratory. Best practices for microscope maintenance. So you want to make sure that you are very, very, very careful and clear and taking very good care of microscopes. In an actual class, we go through these things to make sure that these are done. You should read through these. These are important things to know. Okay, so cleaning with lens paper, not allowing the lenses to contact the slide, protecting the bulb if there is one from being broken, not pushing an objective into a slide, not using the coarse focusing knob when using 4x or greater. That's an important thing that you should absolutely know. Key thing there will be on an exam is the coarse adjustment knob or the coarse focusing knob is only used with 4x and 10x. Once you then get up to our 40x and 100x, you should only ever use the fine focusing knob. You can also use the fine one here, and I often do. I'll use the coarse adjustment knob on the 4x and 10x. And so of course you start at the 4x, use the coarse adjustment knob. Then I also use the fine adjustment knob to get it as absolutely perfect as I possibly can. Then I move up to the 10x, I use the coarse adjustment knob again, and then I use the fine adjustment knob to get it absolutely as crystal clear as I can possibly get it. Then move up to the 40x and only use the fine adjustment knob. And then moving up to the 100x, you want to put a drop of oil before you move to the 100x and then only ever use the fine adjustment knob. The next one, only using immersion oil with a specialized oil objective, so that's the 100x objective, oftentimes. Cleaning the oil from the immersion lens after using the microscope, not getting it on any of the other lenses. Cleaning oil that's accidentally transferred to any other lenses, and then covering the microscope or placing it into a cabinet to be protected. All right, so the next type of microscopy we're going to talk about quickly is dark field microscope. So we were just talking about bright field microscopes. Now we're talking about dark field microscope. So Perhaps you can imagine what that means. Brightfield microscope had the field as being bright, right? Mostly it was very, very light. In a dark field microscope, there is a small opaque disc placed between the illuminator, right, the light bulb at the bottom, and the condenser lens. Remember, that was underneath the stage. And then this opaque light stop, as the disc is called, blocks most of the light from the illuminator as it passes through the condenser on its way to the objective lens. So what we then have is this dark field with light specimens on it. So when we block most of the light from the illuminator, then it produces a hollow cone of light that is focused on the specimen. We'll see an image in just a moment of that. So the only light that then reaches the objective lens is light that has been refracted or reflected by structures in the specimen. So rather than having your eyeball flooded with light and then having the specimen on there bending and moving that light in different directions and showing different colors, instead what we have here is most of the light is totally blocked, and then a little bit of light is let in along the edges, and then what happens is that light is slightly bent, and then that bending toward the specimen is going to just kind of shine a light, a little tiny light on those things, and the light that is then refracted or reflected by the structures in the specimen is going to be what is seen. And so then it ends up looking like the image shows bright objects on a dark background. So let's take a look at that in diagram form. So here we have our light source, the illuminator. This is our opaque light stop right here. And you can see, so some light is going around the edges, and then our condenser lens, which would be under the stage would be here, you know, with the hole going through it. And then our condenser lens then is going to bend this light and get it to the middle right here where the sample is. So then this kind of focal point here, this bent light comes around and goes to the specimen that is being looked at. And then the specimen is going to be refracting, reflecting the light as it's being hit by this light coming around. And then as it's going up, we have our direct illumination block, and then what is scattered is transmitted. We have our objective lens here. This is what's going into the microscope. And that's what is the light, remember, that is bounced around. It's not direct, right? So we don't see a whole bunch of light coming up like we normally would. We just have these little bits of light peeking around the corner, and then these little bits of light are going to bounce off of the specimen. And then, let's see if we can scroll down and see the picture. There's the picture of what a dark field microscope image looks like. So we don't have all of that light coming through like we do in a bright field microscope. What we see is a dark background, and then we have our specimen there that we can see that are lit up just by the bouncing of that little bit of light that's coming around the edges. Dark field microscopy can often create high contrast, high resolution images of specimens without the use of stains, which is very important for using things like live specimens that might be killed by a stain or otherwise compromised. It may not look the same once it's been stained. For example, thin spirochetes like treponema pallidum, the causative agent of syphilis, can be best viewed using a dark field microscope. So you can really kind of see the wiggly spirochete shape of those guys using the dark field microscope. It allows you to have living organisms. So remember, I mentioned that you should know the differences between these microscopes, how they function, why they function like that, and then why we use them. So one of the reasons that the dark field microscope is used, one, it gives great images, high contrast, high resolution images of specimens, but it's also because you don't have to use stains, which is important for live specimens. All right, moving on to the next type of microscope, we have our phase contrast microscopes. A phase contrast microscope uses refraction and interference caused by structures in a specimen to create high contrast, high resolution images. again without staining. It's the oldest and simplest type of microscope that creates an image by altering wavelengths of light rays passing through a specimen. So to create altered wavelength paths, an annular stop is used in the condenser. So remember, the condenser is right underneath the stage. The light is coming through the condenser, and there is an annular stop placed in the condenser. The annular stop then produces a hollow cone of light that is focused on the specimen before reaching the objective lens. The objective then contains a phase plate, phase contrast. It contains the phase plate containing a phase ring. We'll look at a diagram again. As a result, the light traveling directly from the illuminator passes through the phase ring while light, here we are, let me write that down here, through the phase ring while light refracted or reflected by the specimen passes through the plate. Let's see if we can kind of scroll down here. All right, so the diagram of the phase contrast microscope here. We have our light source, the illuminator, and then we have here the annular ring, right? So here is our annular ring. Below the condenser, here's our condenser lens. And then here it says 1, the annular stop in the condenser produces a cone of light focused on the specimen. So our annular ring, again, we have our light coming through here. It's going to create our cone of light coming up to our specimen. The object or the specimen is going to refract or reflect light. So right when the light hits the specimen, it's going to either bounce off or be absorbed or bend it. Then 2, so that's that coming off here, we have our bent or refracted or reflected light. Then we have our objective lens collecting that. Then 3, light traveling directly from the condenser lens and light traveling through here, right here, through the specimen are out of phase when they pass through the objective and phase plates. All right, so we hadn't gotten that far in the words there in the text up above. So we'll jump back up to the text, but here we are. That was number 3. So 3 is here. We have the light that is bent, refracted, reflected, coming up through the objective lens here. And then we have diffracted light and undiffracted light. So let's kind of scroll back up here and use the text for a moment, and then we'll go back to the diagram. So let's see. The light traveling directly from the illuminator passes through the phase ring, while light refracted or reflected by the specimen passes through the plate. So this causes waves traveling through the ring to be about 1.5 of a wavelength out of phase with those passing through the plate. So it is changing the phase. So they were all coming through at the same speed and length, all at the same time. Not really speed and length because it's white light and it all has different wavelengths. But let's say it's all coming through at one big lump. They're all coming through together, all of these different waves of light coming through. And then that changes because of this phase ring. So the light, there's light that travels through the phase ring, and then there's light that's refracted and reflected by the specimen passing through the plate. So they're off. They're not going the same way anymore. They've changed. And so the waves that are traveling through the ring are 1.5 of a wavelength out of phase. They're not in sync. Maybe that's a good thing. They're not in sync anymore. They're not matched up. So with those passing through the plate, because waves have peaks and troughs, right, the ups and the downs, they can add together if they are in phase together or cancel each other out if they're out of phase. When the wavelengths are out of phase, the wave troughs will cancel out wave peaks, which is called destructive interference. Structures that refract light then appear dark against the bright background of only unrefracted light. More generally, structures that differ in features, such as refractive index, will differ in levels of darkness. So let's scroll down here and take a look at the diagram again. So again, we have our light bending, moving through this cone right onto our specimen. Then remember, we have our light traveling directly from the condenser lens down here, and light traveling through the specimen, and they're out of phase when they pass through the objective and phase plates. So some are going to be moving like this. Let's say they were all moving like this for an example, and then those that are up here, so remember right here, let's see, right here, those that are traveling through the ring are going to be about 1.5 of a wavelength out of phase. So if we say these are going through the plate, and then we have those going through the ring, are going to say, let's see, this is a wavelength 1.5 out of phase. So instead, then we've moved up out of phase. Okay, so they're no longer matched up. So this one would be here, and the other one would have been here, but it's been moved out of phase. So then what we can see is sometimes they'll match up, right? So it might match up again, like, well, sort of like this. And then here and here the same, so you'll get something that's very bright. They work together to produce something very bright. Now, if it is not that way, right, if we have one that is like this, and then the wave as it has been matched up, for example, since it's off phase, goes this way, then this is a peak, and this is a trough, and they'll cancel each other out. And if they cancel each other out, then that'll be a dark spot. Okay, so we have our diffracted light and our undiffracted light here. These two different types are out of phase now, waves, these two. And then as they're going through here, then we get our image up here. So for the wavelengths in phase or out of phase, either add together, like we were saying down here, this wave plus this wave, this is adding together, or cancel out each other. So say like this one and this one down here are going to cancel each other out and then not produce that bright of a wave, a light wave coming through. All right, because it increases contrast without having to stain anything, phase contrast microscopy is often used to observe live specimens. Again, if you don't have to stain it, then you can have live specimens. It's also important for certain structures like organelles in eukaryotic cells and endospores in prokaryotic cells. They just show up really, really well in phase contrast microscopy. And here's an example here of phase contrast microscopy. So on the left, this is a Brightfield image, so something that you could see in a laboratory, something that you would see in a classroom, for example. This is using a Brightfield microscope. And then on the right here, you can see this is the phase contrast, and you can really see how the light, for example, in these very bright spots, of course this is debris here, but let's say like right here in that very bright spot, the waves have added together, and then in the darker areas, maybe down in here, they've canceled each other out. So you can really see some differences in there. All right, so the next one is our differential interference contrast microscopes. Here, DIC microscopes, or Nomarski optics, are similar to phase contrast microscopes in that they use interference patterns to enhance contrast between different features of a specimen. So in the DIC microscope, there are two beams of light created in which the direction of wave movement differs. So you're still creating that kind of off phase. In this case, two beams of light are created so that you have this direction being different. Polarization or wave movement is different. Once the beams pass through either the specimen or the specimen-free space, so where there isn't a specimen, they are recombined, and effects of the specimen cause differences in the interference patterns. Again, when, just like in the phase contrast, that's why they're saying it's similar. If we have that addition of waves coming together, then we see brighter areas. If we have that canceling out of each other, we see darker areas. And everything in between. So there's this combining of the beams. This results in high contrast images of living organisms with a three-dimensional appearance. So much more three-dimensional. You can kind of see here, we'll scroll down just a little bit. You can see how this is very three-dimensional compared to something that we could see in a laboratory, for example, in a classroom. This results in the high contrast. These microscopes are especially useful in distinguishing structures within live, unstained specimens. So again, one benefit to using this type of microscope, the DIC microscope, is that you can use live specimens. They don't have to be killed in the process of staining or of heat fixing them. You can have something that's live. You can see this fungus is still alive. Doesn't look alive to us because it's a photo, but it's alive and it can be seen very, very clearly there. Next we have our fluorescence microscopes. So a fluorescence microscope uses fluorescent chromophores called fluorochromes. So they are capable of absorbing energy from a light source and then emitting this energy as visible light. And we've already kind of talked about that a little bit when we talked about glow-in-the-dark things, glow-in-the-dark stars or whatever you want to think of, objects. So you have the fluorochromes, which can absorb light from a light source, and then they emit this energy as visible light that we can see. Fluorochromes include naturally fluorescent substances such as chlorophylls. So chlorophyll in plants is a natural fluorescent substance, as well as then fluorescent stains. So stains can be added to a specimen, different dyes, and then that can be used in the fluorescent microscope or fluorescence microscope. Dyes such as, again, we talk about Texas red, FITC, are example of fluorochromes. And then we have another example here, the 4,6-diamidino 2-phenyl-indole. You don't need to know that, the DAPI or the acridine orange. So just know that there are fluorochrome dyes, maybe the examples Texas red or FITC, and that those fluorochromes are what are used in fluorescence microscope. You don't have to know this long DAPI name, okay? The microscope transmits an excitation light. So it transmits this excitation light generally in the form of EMR with short wavelengths, such as ultraviolet or blue light. So when we're talking about our fluorescence microscopes, they can transmit, right? They transmit excitation light, in such as UV light or blue light toward the specimen. So it's going to be, instead of our little light bulb in the Brightfield, for example, it's going to have UV light or blue light. Then the chromophores will absorb that excitation light and then emit visible light with longer wavelengths so we can see it. The excitation light is then filtered out, again, in part because ultraviolet light is harmful to our eyes, so that only visible light passes through the ocular lens. So this microscope is using something that's harmful, like ultraviolet light. It hits the specimen with the ultraviolet light. The dyes in it, the chromophores or the fluorochromes, absorb that light and transmit something we can see. And then the microscope is going to get rid of that ultraviolet light, filter it out, so that what we see is just the visible light that's passing through this ocular lens. This produces an image of the specimen in bright colors against a dark background, right? Because we don't have a light bulb light. We don't have any visible light going in to the specimen, so it's going to have a dark background. What we have is other EMR, for example, ultraviolet light, that's going through the specimen, coming in the specimen, but then the microscope is going to cancel that out. Not that we would see it anyway, but it cancels that out, and then what we see is this bright color. So fluorescence microscopes are especially useful in clinical microbiology. They can be used to identify pathogens, to find particular species within an environment, or to find the locations of particular molecules and structures within a cell. Approaches have also been developed to distinguish living from dead cells using fluorescence microscopy based upon whether they take up particular fluorochrome. So they're using that to see if it's washed with a particular fluorochrome. If a live cell will pick that up, for example, and a dead cell will not, then you can tell the difference between the two. Sometimes multiple fluorochromes are used on the same specimen to show different structures or features, because some will pick up one fluorochrome and then other specimens or features will pick up a different fluorochrome. One of the most important applications of fluorescence microscopy is a technique called immunofluorescence, which is used to identify certain disease-causing microbes by observing whether antibodies will bind to them. So antibodies, if you haven't studied that yet, are protein molecules that are produced by the immune system that attach to specific pathogens to kill or inhibit them. So you can take an antibody, and an antibody, as it says here, it's a protein, it's flowing through our body, it's produced by our immune system, and an antibody will stick to something, and then it will either flag down immune cells and say, hey, here's the bad guy, I'm stuck to it, here it is, take care of it, or itself will take care of that pathogen. A lot of time it's just a labeler, like a flag, saying here it is, come get them. It can also be an inhibitor. It doesn't actually kill the cell or the pathogen. It can just inhibit it from getting into something. So maybe it'll block the path that it uses to get into a cell and take it over, for example. So these antibodies are used. There are two approaches to this technique. Let's move on here. It says direct immunofluorescence assay, which is DFA, and indirect immunofluorescence assay, which is IFA. So in DFA, specific antibodies, so for example, they say those that target the rabies virus, are stained with fluorochrome. So they're taking those specific antibodies, those little guys that float around in the body, and say, here it is, I got them. And they stain those with the fluorochrome. Let me scroll to here. There we go. Then, well, I guess I missed part of that. So if the specimen contains the targeted pathogen, so if the specimen, right, what's in the container, what has been taken out of a body, for example, to be tested, contains that pathogen that that antibody attaches to, then one can observe the antibodies binding to the pathogen under the fluorescent microscope. So this is called a primary antibody stain, because the stained antibodies are attaching directly to that pathogen. In IFA, secondary antibodies are stained with a fluorochrome rather than the primary antibodies. The secondary antibodies do not attach directly to the pathogen, but they do bind to primary antibodies. So when the unstained primary antibodies, right here, when the unstained primary antibodies bind to the pathogen, the fluorescent secondary antibodies can be observed binding to the primary antibodies. So if we are not able to stain with the fluorescent dyes, if we're not able to stain the primary antibodies, then we can stain the secondary antibodies that then will attach to those primary antibodies. So in both cases, we're getting to the point where we are locating and staining the pathogen. It's just whether or not it's the primary antibody that's going to stick itself to the pathogen or the secondary antibody, which sticks itself to a primary antibody. Thus, the secondary antibodies are attached indirectly, that's the I, indirectly to the pathogen. So since multiple secondary antibodies can often attach to a primary antibody, the IFA increases the number of fluorescent antibodies attached to the specimen, which makes it easier to view. It makes it easier to visualize features in the specimen. So let me scroll down here a minute. We can see the picture associated with that. So an A here, we can see the Neisseria gonorrhea bacterium that causes gonorrhea here, a direct immunofluorescent stain. That's what this looks like after being stained in this way. Here, let's see. This is the direct here in A, right? And then B here is an indirect immunofluorescence stain used to visualize this larva, the Schistosoma mansoni. And it's a parasitic worm that causes schistosomiasis, an intestinal disease common in the tropics. So here, C, an indirect immunofluorescence, or indirect immunofluorescence. The stain is absorbed by the primary antibodies. So here's C, direct immunofluorescence. We have, this is our bad guy here, our bacteria that's in the body. This is our specimen, right, our sample. We have some fluid with our sample in it. And then we have our primary antibody, which is the black Y here, upside down Y. And we take our fluorochrome and we attach it to, by dyeing, our primary antibody. In direct immunofluorescence, then we kind of mix up our specimen, our sample, with these primary antibodies that have been dyed with the fluorochrome, and then they fluoresce. So we see that here. In indirect immunofluorescence, we then take our fluorochrome, our little sun guy, and we dye this green upside down Y, which is a secondary antibody. Then we mix it all up and we have our primary antibody attaching to our specimen, our pathogen or antigen in this case. And then we have our secondary antibody that has the fluorochrome on it. So it's going to light up. And we see that here in indirect. And again, remember the secondary one, the indirect immunofluorescence, is because we can dye lots and lots and lots of secondary antibodies. And a primary antibody may be just very specific for this one tiny location on this bad guy here, on this pathogen. But then we could have several of these secondary antibodies attached to the primary antibody, for example. And then this would really, really brightly glow. And you can see that here and how bright this is. All right, so next we have our confocal microscopes. So whereas in other forms of light microscopy, remember we're still talking about light microscopes, light microscopy. They create an image that is maximally focused at a single distance from the observer. In confocal microscope uses a laser to scan multiple Z-planes successively. So this produces numerous two-dimensional, high-resolution images at various depths, which can be constructed then into a three-dimensional image by a computer. So this isn't something where you're, like our Brightfield microscopes that we see often, where you kind of look down into the ocular and you can look and you can move things around and see it. This is more of a computer-based microscope. As with the fluorescence microscopes, the fluorescent stains are generally used to increase contrast and resolution. Image clarity is further enhanced by a narrow aperture that eliminates any light that is not from the Z-plane. All right, so any light that's not from that plane that it's looking at. So they're very important for looking at thick things. So for example, examining thick specimens such as biofilms, which can be examined alive and unfixed. So oftentimes when we're looking through our Brightfield microscope, we're kind of trying to focus and focus and focus, and we get to what seems to be a chunk of something. Maybe this image here looks familiar to you if you've looked through a microscope and seen something that looks kind of like a chunk of something. And it seems like as you're moving the slide, the stage up and down, you're kind of focusing at different depths. And that can be very confusing and frustrating to try to get something focused in at one single depth. And in this case, we have a biofilm, which is basically a whole bunch of bacterium stuck together, and they create this film, biofilm, around them to all kind of congregate, stay together. And this is very helpful for being able to look at things like that, rather than looking at it through a Brightfield microscope where you really can't get anything into focus because you're just going through those different planes. So instead, this is kind of like I hesitate to say it because I'm not as familiar with them, but like a CAT scan or a MRI, something like that, where it's taking slices, where it's looking at it from different levels, and then it can be put together in a computer, and then you can really kind of see your way around it rather than just shining a bright light on it. I can go up one more time here. So again, it's a confocal microscope, and it uses a laser in this case. So again, we're not talking about just shining a light bulb on it. And it can scan multiple Z planes successfully. Remember the X and Y and then Z. So you can go along. This is a graph here, X, Y, Z. Depth here, the depth plane. And so it can look through these, produces lots of two-dimensional, very high-resolution images. And then it can be, remember, this one includes a computer. The confocal microscope uses a computer that can then put these images together and then come up with one very nice-looking, very detailed image. All right, next, the two photon microscopes. While the original fluorescent and confocal microscopes allowed better visualization of unique features in specimens, there were still some problems that prevented optimal visualization. The effective sensitivity of fluorescence microscopy when viewing thick specimens was generally limited by out-of-focus flare, which resulted in poor resolution. So again, we have this really thick specimen issue. And this limitation was greatly reduced by the confocal microscope. So when we're talking about the fluorescence, right, again, it's similar to the Brightfield where you kind of focus down and focus up and you can't quite get focus on something thick. So then we had the confocal microscope, which gives us those layers that can then be put together, which is very helpful. This limitation was greatly reduced in the confocal microscope through the use of a confocal pinhole to reject out-of-focus background fluorescence with thin, unblurred optical sections. However, even the confocal microscopes lacked the resolution needed for viewing thick tissue samples. So we're talking about some really thick tissue samples. We want to see something in a much more three-dimensional shape, or look at it as in its three dimensions. So these problems were resolved with the development of the two photon microscope. So that's our example there, or shows you why it is that we have this microscope. So we want to see something that's thicker, that has better resolution for really thick samples. The two photon microscope uses a scanning technique, also fluorochromes, and long wavelength light, such as infrared light. So again, not visible light. And we're not talking about the visible light section. We're not talking about shining a light bulb on it. We're talking about scanning, a scanning technique and long wavelengths, so infrared. The low energy associated with the long wavelength light, so the infrared light, for example, means that two photons must strike a location at the same time to excite the fluorochrome. So we have our long wavelength light and low energy. We need only two photons to strike a location at the same time to excite the fluorochrome. So again, we have the fluorescence dyes, and it excites that. Remember when we talked about that, the fluorescence microscopy, we talked about how we use UV light, or in this example, infrared light, and it is shining on the specimen, hits the specimen, and then that changes over into visible light. So here we have two photons of light striking a location at the same time to excite that fluorescence, right, the dye, or not dye, like chlorophyll, something that's already in the organism. The low energy of the excitation light is less damaging to cells. So the low energy, this really low infrared light, remember shorter, or the amplitude is lower. And the long wavelength of the excitation light more easily penetrates deep into thick specimens, so it can really get its way in there. This makes the two photon microscope useful for examining living cells within intact tissues. So we're talking about brain slices, we're talking about embryos, whole organs, and even entire animals. So the use of 2 photon microscopes is limited to advanced clinical and research laboratories because it's incredibly expensive. A single 2 photon microscope typically costs between $300,000 and $500,000. And the lasers that are used to excite the dyes used on specimens are also very expensive. Then it says, of course, however, as technology improves, it might be more readily available. So that cost, as things do, will come down eventually, and then maybe this will be more readily available and more common. All right, next we're going to talk about electron microscopy. So the maximum theoretical resolution of images created by light microscopes is ultimately limited by the wavelengths of visible light. So again, remember, the visible light, if we scroll back up, was in this very tiny area when we're talking about all of the energy that is out there in the world. It's visible is very small. So when we're talking about light microscopes, it's limited by this very tiny window, this small area. Most light microscopes can only magnify to 1000x, and a few can magnify up to 1500x. But this does not to even closely resemble what we can get from an electron microscope, or EM. These use short wavelength electron beams rather than light to increase magnification and resolution. So we have moved from light microscopes, now we're talking about electron microscopes. Okay, so these use electron beams, so we're shooting electrons at a specimen. Electrons, like electromagnetic radiation, can behave as waves, but with wavelengths of 0.005 nanometers, they can produce much better resolution than visible light. So we're talking about incredibly small wavelengths, which can get incredible amounts of detail, much better resolution. An EM, electron microscope, can produce a sharp image that is magnified up to 100,000 times. So really fantastic. Thus, EMs can resolve subcellular structures as well as some molecular structures. So for example, single strands of DNA, we can use electron microscopes to see. However, it cannot be used on living material because of the methods that you have to use to prepare the specimen. So even though we can get some incredible photos, some incredible images with electron microscopes, it's not as useful in some ways for specific things, different applications where you need to have a living specimen. So there are 2 basic types of electron microscopes. There is the transmission electron microscope, or TEM, and the scanning electron microscope, or SEM. So the TEM is somewhat analogous to the Brightfield light microscope in terms of the way it functions. So our TEM is very similar, and you can actually kind of see that here in this picture. It looks somewhat similar in some ways. We've got our little oculars there to the Brightfield microscope. However, again, it uses an electron beam from above the specimen that is focused using a magnetic lens rather than a glass lens and projected through the specimen onto a detector. Electrons pass through the specimen and then the detector captures the image. All right, so we're using beams of electrons to go through that specimen. All right, so we have our figure here, 2.2, our electron microscopes use magnets to focus electron beams similarly to the way that light microscopes use lenses to focus light. So again, we have over here, we have our example of a light microscope. We have our light source, right? It's shining. We have our condenser, which we've seen multiple times now, which is going to kind of gather that light, get it closer to where the specimen is, And then we have the specimen bounces and bends the light. It enters the objective lens, and then that image is going to go up through. We're going to magnify it further using our ocular lens, and then here's our viewer camera, or your eyeball. Then we have over here our electron microscope, in this case the TEM. We have our electron gun. Remember, this is going to be shooting beams of electrons. What we do then is we're shooting the beams of electrons, and then these right here are our magnets. Here you can see electromagnet. So it's going to be bouncing these electron beams along this tube, this long tube, part of the microscope, through the specimen, and then you can see it continues to bounce them to the objective lens, where then the viewer can see them. Okay, so we're using these electromagnets to bend and bounce these electron beams and get them going where we want them to go into the specimen, and then it's going to bounce around through there and then come back out and then give us a nice image. For electrons to pass through the specimen in a TEM, the specimen must be extremely thin, so it has to be sliced incredibly thin so these tiny electrons can then get through it. The image is produced because of varying opacity in various parts of the specimen. So remember, opacity is the opposite of the transmission, so being able to go through like glass, and then the opposite of that, like a stone, for example, is very opaque. This opacity can be enhanced by staining the specimen with materials such as heavy metals. So I guess that's where the rock example comes from. A heavy metal will then make different parts of it opaque, which are then electron dense. So it's like thicker for the electrons to get through the heavy metals. So it's a type of opacity. TEM requires that the beam and the specimen be in a vacuum, and that the specimen be very thin and dehydrated. So in order for this to work, it has to be sealed up in this vacuum because, again, we are shooting electrons in it, and the electrons will just get out if it's not in a vacuum and we don't have these magnets in there to kind of guide them along. The specific steps needed to prepare a specimen for observation under EM are discussed in detail in the next section. So then we have SEM. And then the SEM, the scanning electron microscope, form images of surfaces of specimens, usually from electrons that are knocked off of specimens by a beam of electrons. So this can create highly detailed images with a three-dimensional appearance that are displayed on a monitor. So again, this one, we're talking about a monitor, a screen. You're not putting your eyeballs up to an ocular and then looking in. It's another computer-based one. So typically specimens are dried and prepared with fixatives that reduce artifacts such as shriveling that can be produced by drying before being sputter-coated with a thin layer of metal such as gold. So you can see why the electron microscopes are not useful for living tissues or living organisms, because we have things like dehydrating, we have things with covering it with gold metal, shooting electrons through it, for example. So all of these things are not conducive to living organisms. Whereas transmission electron microscopy requires very thin sections and allows one to see internal structures such as organelles and the interior membranes, the scanning electron microscopy, the SEM, can be used to view the surfaces of larger objects such as pollen grain, as well as the surfaces of very small samples. Some EMs can magnify an image, wow, up to a lot there. So let's scroll down to this image here. So these illustrations compare the components of the transmission electron microscopes and the scanning electron microscopes. So here you can see this is the TEM, the transmission electron microscope, and here the SEM, the scanning electron microscope. We don't have to go through this every single section here, but You can see here we've already looked at the TEM. This is just a little bit of a drawing here, a little bit different of a diagram. We have our electron gun here. Then we have our condenser lenses, our condenser apertures, another condenser lens, another aperture. We have our specimen holder and the airlock. We have then our objective lenses and aperture, then the electron beam coming through, and then our fluorescent screen and camera here. Then the scanning electron microscope also has our electron gun, right? We're talking about electron microscopes, so we have our electrons being shot at the specimen. We have an electron beam here. We have an anode. We have our condenser lens. We don't have multiple condenser lens like we do here. We have our big condenser lens here. Then we have some scanning coils. Again, you don't need to memorize these things. It's just kind of going through the differences here. Then we have our backscatter electron detector here for what's bounced back. Then a secondary electron detector, and then the sample down here. So let's just scroll down here and take a look at these images. So these are electron microscopes. So remember the TEM, this is what you should remember from these. Okay, so the TEM, it has to be a very, very thin sample, has to be cut very, very thin. In the electron beams, the electrons go through the specimen in order to get incredible detail on the inside of things. Very, very small, very, very thin things like organelles or even little tiny membranes. So here, our image A is a TEM image of cells in a biofilm showing the internal structures of the cells because of the varying levels of opacity. So the dark areas, the light areas, and gives us an incredible detail. You can see even the layers in this little section, or the little strands in there. B here then is an SEM image, so the scanning electron microscope image of the bacterium Staphylococcus aureus. So you can see, again, this is the one that gives us the more of the three-dimensional image. It's the electrons are bouncing off of the surface of these things and bouncing back. So we're hitting it, just bombarding it and making it bounce off of every teeny tiny little area possible across all of it. And so then it's going to give us feedback images back of every indent and puff and cilia and every little tiny part of the outside of it, the surface of the structure. So this is what these little Staphylococcus cells look like. Remember, cocci is round, so you can see how round these little bacteria actually are with that scanning electron microscope image. All right, so then it's discussing here a little bit about biofilms, which we'll talk more about in future chapters, biofilms specifically. And then we can talk about our scanning probe microscopy. So our scanning probe microscope does not use light or electrons, but rather very sharp probes that are passed over the surface of the specimen and interact with it directly. So we have these little tiny probes that are going over the surface of the specimen and actually touching it, actually interacting with it. It's not bouncing light or bouncing electrons, we're actually touching the specimen. This produces information that can be assembled then into images with higher magnifications than what we've seen before. Such large magnifications can be used to observe individual atoms on surfaces. So this can get all the way down to individual atoms, the smallest so far. So to date, these techniques have been used primarily for research rather than for diagnostics. So we don't need to get down to that level so much right now, to the atomic level, to do diagnostics. There are two types of scanning probe microscope. We have our scanning tunneling microscope, our STM, and the atomic force microscope, or AFM. An STM uses a probe that is passed just above the specimen as a constant voltage bias creates the potential for an electric current between the probe and the specimen. All right, so we have the probe is going just above it, just above the specimen, and then we have this voltage that's creating a potential for an electric current to go between the probe and the specimen. So if you have studied any electric currents or taken chemistry or possibly physics, maybe it's mentioned, we have this voltage that's created. So it's sending voltage across this specimen, and then the specimen can be interacting or not in creating an electrical current between the probe and the specimen. So this current occurs via quantum tunneling of electrons between the probe and the specimen. And the intensity of the current is dependent upon the distance between the probe and the specimen. So the probe is moved horizontally above the surface, and the intensity of the current is measured. Okay, so it's moving horizontal, sideways, right, across the surface of the specimen, and as it's doing that, the intensity of the current is dependent on the distance between the probe and the specimen. So it's sending this current, and it's picking up information. It's tunneling. We have quantum tunneling, quantum meaning incredibly small, quantum-sized tunneling of these electrons between the probe and the specimen. The scanning tunneling microscopy can effectively map the structure of surfaces at a resolution at which individual atoms can be detected. So it can really get in there and get a nice, really solid surface map of something. So again, not inside, but it's a surface thing. Similar to the STM, the AFMs have a thin probe that is passed just above the specimen, so in that same way. However, rather than measuring our variations in the current at a constant height above the specimen, an AFM establishes a constant current and measures the variations in height of the probe tip as it passes over the specimen. As the probe tip is passed over the specimen, forces between the atoms, if you remember from chemistry, the van der Waals forces, the capillary forces, chemical bonding between atoms, electrostatic forces, and others, cause it to move up and down. So remember the STM, you have the probe that's going at the same height across the specimen, sending voltage to measure the currents as it goes across. In this case, we have a constant current, okay, rather than measuring the currents, we have a constant current, and it's measuring the variations in the height of the probe tip as it goes over the specimen. And as the probe tip is going over the specimen, it's going to be picking up on the differences, like the measurements between the different atoms. So it's going to pick up on the forces, these minuscule little forces that are going on within atoms and between atoms, and that will cause that probe tip to move up and down. Deflection of the probe tip is determined and measured using Hooke's law of elasticity, and this information is used to construct images of the surface of a specimen with resolution at the atomic level. So we're looking again at the atomic level in these cases. So here are images of that. The STMs and AFMs allow us to view images at the atomic level. This is an STM image of pure gold surface. Shows the individual atoms of gold arranged in columns. These are individual atoms that we're seeing. And then in B, we have an AFM image that shows long strands. Those are molecules of nanocellulose, in a laboratory-created substance derived from plant fibers. So we can really see things. We're talking about nanoparticles there, nanocellulose. So down here, I just want to point out, we're not going to go through all of this. We've already talked about each of these things. But here we have these wonderful charts. These are the light microscopes. They tell you the magnification, which you should know. This would be incredibly helpful for you to study. We talk about all of these different things. It puts it in a nice chart. The uses, this is what you need to know. So you need to know whether it's a light microscope, the general magnifications possible, what it's using. So this is what a light microscope is, using visible or ultraviolet light to produce an image. The different types, so bright field, dark field, phase contrast, etc. The uses of these different types, and then some sample images. What kind of image we are getting back, okay? And then as we move along, it keeps going. All of the other ones we've talked about, more light microscopes, you should know all of these. And then we get to the other ones we talked about, so our electron microscopes, the EMs, the magnifications, the different types, TEM, SEM, the uses for these, and then types of sample images, meaning what we're going to get back. Remember, TEM are lots of internal structures, very thin. SEM, we're looking at the external surface structures. And then the last one that we talked about, or last ones, the scanning probe microscopes, the STM and the AFM, and their uses, magnifications, and what a sample image would look like. So those are all things that you should know that we just went through from this section of the chapter. So knowing the different types of microscopes is part of microbiology, part of learning about microscopy, and you should be able to remember each of those things and understand what they're used for.